Let's see how this works then. So neuromuscular junction. So a little recap here. Gives a nice 3D shape. So here we've got our friend motor neuron. Inside we've got some vesicles, acetylcholine, little channels here, which, remember, quick recap, are calcium ion channels. These are voltage gated, and what we've got here is an interaction between a neuron, a motor neuron, and a muscle cell. Okay, so we'll a bit more overview on a muscle cell in a moment, but very simply this little animation shows what happens. We get depolarization of this membrane here, causes calcium to enter, run it through, so action potential there, calcium channels open. Remember, those are voltage gated calcium ion channels, key term there, ion. They're diffusing down electrochemical gradients. In turn, that will cause these vesicles to migrate and fuse with the presynaptic membrane. In this instance, they've released their neurotransmitter, these blue little acetylcholines, into what will be the synaptic cleft. At this point, we call it a neuromuscular junction because we're not going to another neuron. We're actually going to this membrane here, which is a continuation of the outer membrane, which we'll just see in a minute when we pull out, of the muscle cell. If you remember, that was called the sarcolemma. Lots of new terms in this topic. So the sarcolemma. So the idea is this will migrate across. Feel free to pause this at any point. We can see the acetylcholine binds. These are ligand gated sodium channels. So okay, this is diffused across the gap, the synaptic cleft, but we'll call it the neuromuscular junction, and it's caused those channels to open. Sodium's entered, and that will depolarize the membrane. Now at this point, the membrane isn't a postsynaptic membrane, it's this membrane here, which just follow my cursor's gone, there we go, which is moving around. Again, three-dimensional structure. This is the outer membrane of a muscle cell. So this is your sarcolemma membrane, and you can see that the action potential has passed from this membrane all along there, sodium, to this region called a T-tubule, basically a transverse tubule. It runs into the muscle cell from the surface, so it's a continuation of the sarcolemma and down. So what happens is this depolarization passes down and causes this yellow structure as it's labelled here, the sarcoplasmic reticulum. And again, sarco just prefixes muscle. So in effect it's an endoplasmic reticulum, and its job is basically to store calcium ions. Now when this depolarization arrives, what it does, it causes depolarization of this sarcoplasmic reticulum membrane. That opens calcium ion channels and allows calcium stored within the sarcoplasmic reticulum to flood into the sarcoplasm. If you remember, the sarcoplasm is this region inside the muscle cell. We see that the calcium's coming out, and the job of this calcium at this stage, we'll get into more detail later on, is to cause this stripy structure, this myofibril, to contract. These are the, if you remember, we said that the muscle cell is like a packet of spaghetti, and each strand of spaghetti is a myofibril. Here's one of those strands of spaghetti, and the idea is this calcium will cause this to contract, to get shorter. And a quick mention here from the diagrams we've already looked at, this dot line here and this dot line here are referred to as Z lines or Z discs, and if you remember the distance from Z disc to Z disc, or Z line to Z line, or Z line if you're American, this here is one sarcomere. So we've got sarcomere, sarcoplasm, sarcoplasmic reticulum, and sarcolemma. And if we just watch this latter part of the animation, we'll see that this Z line to Z line gets shorter, sarcomere gets shorter, all is grand and good. There we go. And the neurotransmitter there is reabsorbed, acetylcholine is resynthesized. Excellent.